All right, so good morning. Welcome to the second week. And we have got a fresh lineup of speakers for you this week. And the first to start would be Dr. Amitabh Nandi of the Department of Physics, IIT Bombay. So again, Amitabh has had a long association with IMSC, actually from the time you were a student, right? At Jawaharlal Nehru University with Ram Ramaswamy. So Amitabh had been coming to IMSC and giving talks on his various works. In fact, I still remember the time that we discussed the stochastic simulations, how to adapt, I think, the stochastic simulations to the kind of kinase reactions that we were doing. And so over the years, it's been a pleasure to discuss a lot of things with Amitabh, and so it's with great pleasure that I invite him to now give this talk. So this will be the first of his two talks. The second talk he'll be giving tomorrow. Okay, Amitabh.
Okay, thank you, Shita bro, for this very generous introduction. And I would also like to thank you and the other organizers, Shakti is here, Mithun, for the kind invitation to talk about my work and also discuss in general some of the problems which we had been discussing in this workshop. So what I initially planned that I will talk about, like give like an overview on morphogenesis by taking two different examples at two different stages of development in Drosophila. One is looking at a very early stage syncytial embryo, and the other is looking at the fly-wing epithelia. But I think I won't have much time to actually cover both of these problems, and this problem on syncytial, studying the syncytial embryo is rather new. So what I will do, I will mainly focus on the wing, like remodeling the wing epithelia problem, which is rather old. It has been a while that we worked on this problem, but I will focus mainly on that, and if time permits, I will discuss about the syncytial embryo problem. So the main idea what I want to discuss is how like a piece of tissue is remodeled, a shape emerges due to the collective interaction, due to the interaction of many individual cells. So how one gets like a large scale macroscopic property of a tissue due to collective interaction of individual entities. So I will try to give some broad overview before I jump into the problem, and if you have any questions, please stop me right there, and please feel free to ask me any question. So let me start with a quick discussion on this very seminal paper by Phil Anderson. More is different. So the main idea of this paper was the following. So he said that if you have a collection of small entities where you exactly know the physics of these individual entities, then the interaction between this entity leads to large scale macroscopic properties which cannot be thought of by, like, you cannot just simply extrapolate the individual properties of the individual entities and get those macroscopic properties, and this property emerges out of the collective interaction. And one can think of several examples in physics. For example, I am showing here a schematic here. So you can think of the conventional liquid gas transition where you have individual molecules, and if you keep on changing temperature, because of interaction, you see suddenly a change in phase. You go from a gas to a liquid or a liquid to a solid. You can think of more exotic interactions like something giving rise to superconductivity or superfluidity, which are known in, which are very well studied problems in physics. And these are all equilibrium systems. Basically, the system is connected to some temperature bath. Now, if you are a statistical physicist, the main problem in this here in this context is you are looking at the interaction of many small entities and see how a macroscopic behavior emerges. So as a statistical physicist, what your main goal is to calculate this quantity. This quantity is also known as a partition function. So you see this curly H, and this curly H contains all the information of the microscopic rules of interaction. Now, if you are able to calculate this, in most of the cases you cannot because of the complexity of the problem or the kind of interaction between these individual entities. If you are able to calculate this, this quantity, then this is your gateway to macroscopic properties of the system. The simplest example is if you have a partition function for a simple non-interacting gas, you can derive the macroscopic thermodynamic laws which are well understood. But as I said, in most of the cases it is not possible. Then people find out different ways to tackle, either construct toy models or like simpler models to construct this and see what kind of macroscopic laws emerges, and or one uses some other techniques which is like constructing a phenomenological free energy and whatnot. Now, the main point I wanted to discuss this is because now the problem I discussed here, think of a generalization of this problem. This was an equilibrium problem. Now, think of a more complex generalization of this problem in the context of biology. So, at very small scales, at the scale of nanometers, you have protein production. Okay. Now, these proteins, you can think of force generators or controlling various metabolic activities. Now, these proteins are important to ensure that you have a properly functional cell. Now, you think you go to a higher scale and you have many such cells, and the cells are interacting with each other. And as a result of this interaction, you see emergence of more complex shapes. So, if we are able to put an arrow this whole process starting from a very small scale to a very large scale, can we think of life as an emergent phenomena? I mean, I'm just putting it a little naively because it is too far-fetched, but what I'm trying to say is, can one see development of an organism or like the process of morphogenesis as an emergent phenomena which comes because of there are different scales, and at every scale there is a broken symmetry. And just to again give you an example, I show you the development phases. It is a time-lapse movie of zebrafish which starts from 4-cell stage, and you see all the cells are dividing, and they keep on dividing until something is triggered and they start collectively flowing. So, this forms the tail, this forms the head, and eventually you see emergence of these nice patterns which forms the somites, which eventually will become the vertebrate. So, if you think of like, say, I want to focus on one small part of this tail which was emerging. So, I can take like such a snapshot and look at the movie. So, these what you see are all the nuclei which are moving in the tail bud, and you see really collective flows of tail. Now, I can ask the same question. So, there are various scales in this problem, and if you are interested what is happening inside the cell, there are a lot of proteins. They are interacting with each other, and these are the proteins are very small in number, so they are very fluctuating quantities. And how would you study such processes? A correct way to study such processes is to write down a time evolution equation for the probability that you have certain configuration of proteins, and one can write down such an equation, and this is known as the master equation. So, basically this is a time evolution equation or a conservation equation for the probability. So, one can write such an equation and look at the stochastic time evolution. Now, if this evolution is not stochastic per se, so you think that the number of proteins are large, so then you don't need such stochastic evolution. So, what you can do is you can calculate average quantities of these proteins, and if you calculate average quantities, so those will be some dynamical equations for the proteins interacting with various other proteins, and this is where this whole notion of dynamical systems or nonlinear dynamics comes, where you can use those to see how this whole network of proteins will evolve. However, if I think that I am not interested in these processes, the proteins, how they are interacting, but I am interested in really the slow scale dynamics of how this shape emerges. Okay, so if I think that, so this shape, if you think this emerges over a span of roughly 24 hours. However, if you look at how the proteins are interacting, these are at a very fast time scale. So if you are interested in this really slow time scale dynamics, what you can do is, you can take like a big box and say that, okay, I will not worry about what is happening inside each cells. I will worry about that how each cell is interacting and how this leads to some formation of shape or collective flow. And in that case, what you do is you do something called coarse graining, and instead of looking really the first time scale processes, you worry about the density, or if there are some other macroscopic variable, here for example polarity, because they are moving in some direction, and then you try to write down some coarse grained equations. And I have written down a representative coarse grained equation. It does not necessarily like explain the particular mechanism I'm showing, but just to show that one can write down this kind of coarse grained equation which gives you the evolution of the density, or if there is some other observable which is important and really helps you to understand the slow time scale behavior. Now,
what exactly are the various symbols over there? What is C, what is P?
So again, this is just a representative
that,
equation. So what I'm saying is, in the microscopic picture, I have say n cells.
Yes.
And each of them had some polarity P. So what I'm doing is I'm coarse graining these. So I have a density field which is C, and I have a coarse grained polarity field which is now continuously varying in space. So the first equation is simply the conservation equation for the density, but it will not be conserved if there are cell divisions. So that's why or cell death. That's why I have put a term S naught over there. The second equation gives you a time evolution for the polarity. So again, the second term on the left hand side, it is some kind of an advection term for the polarity. And on the right side, I'm saying that there are some properties of the polarity field which can be derived from a free energy. So this is that gradient of free energy, and if there are some fluctuations, that is this fluctuation. So I have just written, although it does not represent this system, like the Tonat2 model in some sense.
Sure, sure. And how would you define polarity in a general context? I mean, for a specific context, I can understand that you can define polarity, but in general, what would you call polarity? It's just a order parameter, or?
In this case, I'm thinking of an order parameter, say, if individual cells has some sense of a directionality. So if I know the instantaneous directionality, I will call it a polarity. I mean, typically there are cells where there are proteins which are redistributed asymmetrically, and that define a polarity of these cells. So there the definition is, I guess, more intrinsic. So I am thinking of, there is a polarity which is already defined at the microscopic scale. I am just coarse graining this and defining a more macroscopic polarity field. But because of certain asymmetry in the protein distribution at the microscopic scale, there is an existing polarity.
So are you taking space and time as variables and checking the concentration of proteins as they vary in space or time, or
here,
cells in space or time?
Okay, so here I am just taking this, in this context, say the number of cells as the density. So I am not really looking at the how the protein concentration changes or anything. Those information, like the details of how the protein regulates the polarity, those will be hidden in the coarse grained parameters, for example, the lambda. So when we do that, it's a very good question, because when you are interested, say, let's go back one step back. So here we were interested in the time evolution of the proteins. So there we were really tracking the time, how the proteins are evolving in time. Okay, but now I am saying that I am not, like, I will not worry about these fast time scale processes where this protein changes. I will think of overall how the collective interaction between the cells are happening, and any information regarding those fast time scale processes will be hidden inside the parameter. a simple analogy you can think of like say a fluid. The fluid is made up of individual constituents, such molecules, so they collide with each other and whatnot. So there are some intrinsic properties. But when you see at a macro scale, like a fluid, you have a single macroscopic parameter, which is the viscosity. But the source is actually the interaction at the microscopic scale. Okay. Okay. So
equilibrium, or in general you are kind of assuming some local thermodynamic equilibrium, because you're writing down a free energy. I imagine these systems are not in equilibrium.
The system is not in equilibrium, but there are certain equilibrium properties, so the non-equilibrium part enters here. But if you think there was no non-equilibrium part, so there is like, think of like, then this system is kind of like a moving magnet. Okay, so then you can define a free energy. So you are saying that there is one part which is purely the passive part, which can be derived as a gradient of the free energy, but the non-equilibrium part is still there. So the system is essentially out of equilibrium.
So you are assuming some local thermodynamic equilibrium. Is it? And because see, if it's not in equilibrium,
I am not assuming local thermodynamic equilibrium. I am saying that if you think in the continuum picture, you have forces which are generated. One force is generated purely due to interaction between the entities. That does not have any non-equilibrium part. Even if there were no non-equilibrium forces, it can be derived from a heat bath. I am writing that part separately here, but any non-equilibrium contribution will come separately, which is here. So the system overall is out of equilibrium. Okay. Yeah. So I will go slow. Maybe I'm not able to finish this talk, but I hope like it is okay, and we can continue maybe after the talk also. So this is a very seminal book by Darcy Thompson, and some of you might have seen. And the idea was, he's a Scottish mathematical biologist, so he actually already back in 1917 thought about that during morphogenesis, the different shapes which are formed, there are underlying physical laws, and there are chemical and physical laws, and one has to use mathematics to understand this system. And it's a very beautiful book and very well written. So, for example, I quote like something from this book. The problem at that time was there were no good experimental ways to quantify that what are the physical mechanisms which are happening, how actually things are moving around in the cell, or if you take a piece of tissue, you couldn't put under the microscope and see the cells are moving or interacting, or what kind of forces are generated. So these ideas were not was not able to there was no way to verify these ideas. But now you know, with the advent of fluorescence protein, you can tag something and actually see under the microscope how things are moving, and in today's day, these ideas are extremely relevant. So what happened, like if I see a progression, so during Thomson's time, because there were no way to quantify the physical aspects during development or in biology in general, so there was a huge development in this field of molecular biology, where people started looking at the different interaction pathways and quantify like what are the different protein networks or signaling networks, and a lot of work has been done. But one big breakthrough came during with this paper by Alan Turing in 1951, which is the following. And everybody, so I think during this meeting, this paper has been discussed quite in detail, so I will not get into these details of this paper. So the main idea was, you can have these chemically interacting species which are morphogen, and now you don't have a homogeneously mixed system, you have some interaction, and now if you allow diffusion of these different morphogens, so here I show a schematic of a particular morphogen, which is the activator-inhibitor system. Now this interaction plus diffusion with different diffusivities of these molecules in potentially can give rise to this kind of patterns. So this was the main idea, and it was a remarkable idea. But there are certain things one should take into mind, because if you think of achieving like development of an organism simply by this kind of reaction diffusion processes, there will be some caveats, because diffusion is a very slow process. Okay, and I will give you an example. And in principle, these are not simply proteins which are diffusing. There are mechanical force generators inside the cells and tissues which actually speed up the process of redistributing these proteins. And these are basically something chemomechanical processes or mechanochemical processes. In other words, you have chemical processes which generates forces, and these forces actually drive transport or flow, and that can redistribute the chemicals. Let us see like some example. Suppose a simple example, I have some molecule which want to start from here and reach this destination. If it was just simple diffusion, I know how much time it will take roughly, if I know roughly the distance, if I know the diffusivity. So I can make some estimate of time. So if it is like an E. coli cell, the radius is roughly 1 micron, so it will take around 10 milliseconds. If it is a bigger cell like Dictyostelium discoidum, it might take even longer, it will take roughly 42 seconds. But if it is a long axonal cell, like a neuronal cell, so it is going to take 20 days. So clearly, there is a problem if it was simple diffusion. And all of you know that it is not diffusion inside cells, because there are these force generators which are called molecular motors. So they can hydrolyze ATP, and they use this released energy to do mechanical work. So what they do, they can work on these microtubules. These microtubules have a polarity. So there are proteins which are these kinesin proteins which can work from minus to plus, and dynein which work in the reverse direction, and while walking they can carry some cargo, and this can actually, they can walk with a constant speed. Okay, so everything is over damped, so they will walk with a constant speed. There is no acceleration or inertial terms. So all of you know about molecular motors and stuff, right? So again, to give you like a ballpark number, so typically a motor will walk with a speed 1 micron per second, and a typical protein diffusion coefficient, if I take like 10 micrometer square per second, if I am traversing a distance which is d by v, which is 10 micron, this motor will be advantageous, any kind of motor transport. Otherwise, if we are traversing very short distances, it is still okay to use diffusion. And there is another kind of collective flow which is induced by myosin motors, and how it happens, and this is actually like very interesting. So you can think, so if you look at this small schematic, this is a network of actin filaments. Now if you zoom, you will see that it is a network of actin filaments with cross-linking myosin motors, and these myosin motors again can work on these actin filaments, and they can generate, like, create local force dipoles. So I have a cartoon, I guess. Yeah, so it can, so typically if your actin network, it also has a polarity, and they are typically anti-parallel in nature, and when the myosin motor works, they generate some kind of a contractile force dipole. Now because there are many such actomyosin contributions, if you coarse grain and if you average over, you will get some kind of a stress field generated. And if you look carefully, this is one such scenario in C. elegans, where what you are seeing, it looks like a fluid, but it is actually a network of actin and myosin, and you actually see a flow from this direction to this direction. So it might not be immediately clear. For example, if you quantify the flow field, like using something called particle image velocimetry and plot it, you will see there is a non-zero velocity field in the negative x direction. So the point is, and this now should be coupled with this idea of Turing. How? Because this is a mechanical mechanism, there is a force generated in this fluid. So you can think that what this equation means, again, this equation actually solves for the velocity field. So this quantity is a stress field. So you right now, those who don't know don't have to worry about this equation. So this equation is simply saying that what are the different forces that is balanced in the continuum limit. So if you solve this equation, you will get a solution for this force field, the flow field, the velocity field. Once you have the velocity field, you had the original Turing's idea of reaction diffusion system. You can put this in this thing. What it means is you have diffusion of these proteins, but because there is a flow created in which these proteins are suspended, this flow will take this, advect these proteins faster. So they will redistribute these proteins. And this is an essential ingredient in morphogenesis, because morphogenesis, if you think of like the whole organism that develops over a span of like several hours or over one day, if you don't have this contribution, it will be rather a slow process. Huh. What is the? Okay. So this is basically, I think you had been introduced to this term during this workshop. This is basically how the chemicals are reacting with each other. So this C, it is composed of several proteins. It can be X1, and each of these, if there was no diffusion and advection, each of these has so this is basically the dynamics. So how these proteins are interacting with each other. It's a set of coupled ODs.
In this context, like in the actomycin context,
in this context, it depends what proteins are there which is embedded in this actomycin network, which are redistributed. For example, in C. elegans, there are these proteins called the PAR proteins. So these proteins are advected and transported using this flow field. So it will depend on how these PAR proteins interact with each other. It depends on the system. It can be in principle any protein.
Thanks.
So this is basically in principle, the dynamics part, or the nonlinear dynamics part if there are nonlinear interactions, and the rest is purely the diffusion part and the advection part.
So if I didn't have the advection term, would I still get eventually the same state, except that it would occur very slowly, or is it that the advection term would qualitatively change the pattern that I would get?
I think the advection will also have a contribution to the nature of pattern you get. See, one big difference is diffusion is a symmetric process. I mean, if you have a localized this thing, it will symmetrically spread. Advection is a more directed process. So depending on the system, I guess, I mean, the nature of patterns also which you get, that will change. I don't have immediately some examples.
So it's not like the stirring of sugar in a cup of tea, where the stirring is just basically making the process faster. It doesn't change the eventual end state.
Yeah, so, but sugar is, you have only one component. Yeah. I mean you need multiple components. So the C is a vector.
There is no reaction there.
Yeah, there is no reaction there.
And I was curious, what exactly is this picture showing, the movie that you're running? What are those bright spots?
Yeah, good question. I think these bright spots are these myosins, which are very clustered in the actin network, if I remember it correctly. So what actually you are seeing is, I mean, you cannot resolve the individual myosin motors because of, the resolution. But what you see is there is a collective flow which is generated, and the reason it is generated is this whole actomycin network, it has a gradient. So in this part, the gradient is stronger. There is more contractility, whereas close to here, the actomycin network is weaker. It has less contractility, and because there is a single connected fluid, I mean, you should, although I am saying actomycin, it looks like it is some filamentous network. You think of this in the long time scale mode, so where you can think of this as a fluid. And because there is a single connected fluid, the gradient contractility or gradient stress, it generates a flow in one direction. Okay. So again, so here I have tried to give this example that these are actually mechanochemical processes.
Can you explain just once again how the force balance is being done?
This force balance equation, right? So what I have done is here, so so anybody knows the Navier-Stokes equation here? So basically I will tell you. So the Navier-Stokes equation is some kind of like a Newton's equation for a continuous flow. Okay. So on the left hand side, you have mass times acceleration. Of course, when you write acceleration for a fluid, you have to take into account several things. So I'm not getting into. And then here you write the sum of the forces. Now, on this side, so the sum of the forces, one force is derived from the stress field. So which is basically in continuum limit, you write the stress as a rank 2 tensor. That is what has been written. And you derive the forces as a gradient of this tensor. Okay, and this is what it is. So this is a force, the ith component of the force. Now, the fluid, this cellular fluid is a highly viscous fluid, so it cannot accelerate, which means, so in other words, it is also called as like a fluid with very low Reynolds number, which means that you can drop this term. Okay, so what you have on the right hand side is this, plus if there are any other forces, and that force is a dissipative force, which is due to friction, because you have a substrate, like you have a background on which, and this is what it is.
This is the reaction diffusion process with advection.
This one?
The second equation in the slide. Yes.
Oh, here.
Yes.
So you are okay with the first equation, right? This is the dynamical part, which I have explained here. So this is basically the interaction between the proteins. You cannot see those proteins, they are say somewhere over here. Now these proteins can diffuse. This is the diffusion part, and they can also be advected. And this advection is happening because overall the background fluid has a flow. So this is what this equation means.
But like, I could not understand why we subtracting the advection term.
Oh, that is basically a convention. So one can see while deriving. It is not subtracting. So if your speed is in the positive direction, you get a negative sign.
Okay.
So this is just, I mean, while you derive this, you will get this.
Okay.
So it depends on, so the V has a directionality here.
Right.
Okay. So I will try to wrap up this part. So this was a longish introduction, but you know this reaction diffusion plus
Sir.
forces generated.
Sir. In the first equation, when we were force balancing, we basically removed the first term because we said that there will be like no net flow or something like.
No, I'm saying that there is no acceleration. The fluid cannot accelerate.
Okay, so there will be flow.
The damping is very high.
Okay, so there will be flow, but the acceleration is not there.
There is no acceleration. It cannot accelerate. Okay. See, this kind of force generation, so molecular motors were not discovered during Turing's time. But yet, he actually knew this. So he had already mentioned in the paper that there is two parts. There is a mechanical aspect also, but it makes the problem more complex. So for the moment, he will ignore it. And later, people found that there are these force generators which are molecular motors. Anyway, so let us come to this problem. Now, I think I have given you kind of an introduction where hopefully I have convinced you that if I want to study this whole developmental process, I have to take into account the active forces. So simple diffusion is not sufficient, and one can think of morphogenesis as an emergent phenomena in the sense that you have collective interaction which gives you very large scale remodeling and self-organization. And here again I show two examples. So without losing much time, let me come to the problem in hand. And what I will study is a very specific problem which actually we studied quite some time back. I hope I remember all of it. And if you don't understand anything, please stop me and ask. And I will give you more of a geometric picture without going into equations. So the idea is how this wing shape emerges due to various things that are happening at the level of this wing at a very early developmental stage, namely the pupal stage. So this work was a collaborative work with some very fantastic people, and all the experiments were done by Raphael in Susan's lab, and from the theory side, like all these people were involved. So let me start by asking a very general question. Suppose you have a piece of soft material, in this case a tissue. Now suppose this material is under tension because of forces at the boundary. You have put some boundary conditions. Okay. Now because it is a tissue, and this is like a tissue in a living organism which is developing, now you know that there will be also stresses generated at the level of individual cells. And the reason for this is, I have just shown that there is this network of actin and myosin close to the cell boundaries, and they can generate stresses. Now if you think that each of these individual cell is like an active material, so you have now competition between two kinds of forces. You have stresses generated at the level of individual cells, and you have forces at the boundary. Now how this interplay is going to give you some emergence of shape for this system, and can we understand that by looking at this Drosophila wing and quantifying the mechanical properties of individual cells and the tissue as a whole. So this is the question, and to understand that, what we will do, we will look at how this wing structure, starting at 16 hours after the puparium formation, over a span of 16 more hours, emerges to a shape like this. You see that there is a drastic change in the shape. So let me quickly give you some overview of this piece of tissue. So this is an epithelial tissue. It is like a two-dimensional tissue. It's a single cell layer. Actually, it's a bilayer, but you only look at the single layer. It has two tissue types. One is the hinge, which is attached to the body. Another is this distal part, which is the blade, and colored with different colors, and they have different cell properties. And as you see over time, over a span of 16 hours, this hinge has shrunk quite a lot, whereas you might not be convinced, but I will convince you eventually that this blade, the area has not changed. What has happened, it has changed its shape anisotropically, which means it has extended in the x direction, but at the same time contracted in the y direction, keeping the area fixed. And the question is, the same question, what are the different players here, which is giving rise to this? And let me now show the actual movie from experiment, which shows you this process. And it's amazing to just stare at this movie. So you can see these polygons. Each of these polygon is a cell, because the membrane proteins at each cell has been labeled with some fluorescent protein. The cadherins had been labeled, so you can resolve the deformation of individual cells. And as you keep on staring at this, you see, let it start again, there is a huge collective flow which happens from the right to the left. It starts moving in this direction, and you can see, and eventually this shape goes from this to this. You see formation of the veins and all. These are much complex. Now, over the span of 16 hours,
Is the thickness affected over the course of development?
the thickness,
Thickness.
no, so it's a single cell, it's a single cell layer. So you are saying whether the volume, how the volume is modulated, good question. I don't know actually. So in principle, whether the volume remains unchanged, and yeah, I actually don't know. But experimentally, is it possible to quantify? Probably yes. Yeah, I don't know. So it is, the actual thing is like a bilayer. You have one layer which we are looking, there is some other material, and then there is another layer which you don't see. But we are concerned with the one layer, but nevertheless your question is still valid because each layer has a cell which has a volume, a height. Whether that is also modulated, yeah.
So at the periphery of the wing, is there something else other than cells?
Yes, I will come to that.
Okay.
That is an interesting question that goes back to my first hypothesis that the thing is mechanically under tension due to boundary conditions. So I'll come to that. Yeah.
At the initial stage of formation, it seems to be that there are some foci of high cadherin concentrations, and it also seems as if some cells are like sort of losing their cadherin signal. So is there some degree of apoptosis, and
Yes.
is there some degree of like
Cell division also.
some degree of what you would call predetermination that these regions should always have like high expression of cadherin so that the wing sort of develops in a normal way?
So that's a very good question, and I don't have a very clear answer to that. Those are more complicated processes, and you are right. So probably there is already some pre-existing cues which, like at some located points, for example, how the veins are formed. So for those, these cues might be important, and I don't know. So what can be done, I will tell you. And coming to this other question, there are apoptosis. There are cells which are dying. There are also cells which are dividing. Okay, so there is both. And both are important. And so let me explain, and again maybe, yeah, sure.
There are curly wing or small wing variants of flies already, I think, in Bloomington. So can you sort of compare? I mean, the fly stocks are available. So can one compare like normal winged flies, wild type wing sizes, and small or curly winged flies to sort of get an idea of how the mechanical processes explicitly pattern the wing?
Clearly we haven't done it, but I will talk to you more to
Sure.
understand like about like how to make this comparison and like what one should look at.
Sure. Yes,
Yeah,
thank you.
yeah. Okay, good. But let me quickly explain what you see here. So you see these collective flows and a remodeling of this piece of tissue. Now, it is remodeled again due to various things. One is what he has pointed out, there are cells which are dying. They just exclude themselves from this network. Then there are cells which are dividing. Then the cells are exchanging neighbors also, okay, which is a crucial part I will come to. And there might be some cue here which is driving this large scale collective flow, that they tell you that there is a stress gradient due to which they have to move collectively from one direction to another. There are a lot of things, and you have this formation of the veins. But now, overall, what we want to do is address a very simple question. So there is a lot of things happening here. The question we are going to address is simply, I see this shape, can we use a very coarse-grained, simplified continuum model to understand how it will go from this shape to this kind of a shape? And for that, what we want to do, we want to quantify whatever information is there at the level of this wing. So, because we want to extract some mechanical properties from this experiment, and this is a beautiful experiment, and sorry, yeah, so this is the question, and the first thing we should do is segment this movie. So this is the experiment which was done under the microscope. What you can do is you can generate a computer version of this movie where you have exactly the pixel, like the polygon information, where you exactly know how each polygon looks like. And the experiment itself is beautiful because it can track more than 10,000 cells over a span of like roughly 16 hours, which is a great achievement. So once we have this segmented movie, we want to characterize two things. Overall, how much deformation of the tissue has happened and what on average each cell deformation looks like. Because if I think that I will finally want to quantify how much change in this whole thing is happening, it is due to like whatever is the cell shape change over time due to whatever reasons which we will find out, and boundary conditions which I have not come to. So let us try to quantify this, movie. So think of this schematic. So I have like a network of cells with the cell centers, and if I have this movie where I can track each of these cells, I can calculate the spatial velocity field, right? Velocity field as a function of position. And if now I calculate the velocity gradient from this calculated velocity field, I know that the total tissue deformation is related to the velocity gradient tensor. How? Just to give you an example, I should integrate the velocity gradient tensor over all time, and that will give you the relative change in the dimensions. For example, here I am showing a small example. You started with some length of this box L, and it has extended only in the x direction by delta L. So the change, the relative change in this length is simply the velocity gradient integrated over time. So I can quantify the whole tissue change from this. How should I quantify on average how each cell deformation is happening over time? That is a little more complex, but thanks to Matthias, he found a very nice way, and the idea is the following. So you have this network of polygon. What you do, you construct a dual network by connecting the cell centers, and the dual network of this polygonal network is a triangular network. So now you can ask that how each polygon has changed from some reference polygon, which is an equilateral triangle. And I'm not going to go into the details of this method. So there is like a bunch of papers which they have written in characterizing this kind of cell elongation from this network dynamics. But the idea is, if you take a regular like a triangle and see how the change has happened, like take any triangle and quantify the change in this dimension with respect to a regular triangle, that will give you a Qij for each of these triangles over time. And what we can do is, so now with this, we have the cell elongation or the cell deformation quantification. So now we can compare these two. And again, I told you right at the beginning that because the main deformation is anisotropic, so we will for the moment focus only on the blade. Okay, so here we can go a little slow because there is a lot of interesting things, results that will come. So because we are going to focus only on the blade, and if you recall, the blade was extending anisotropically, means it was extending in the x direction. At the same time, the deformation in the y direction was equivalent, so that the area was unchanged. So I will compute what is the overall change of this anisotropic extension of this blade, and on average how the cell shape is changing inside. Okay, so for computing the anisotropic deformation, I should look at this. Now this is probably you already know, if I have this deformation tensor which is rank 2, I can decompose it into two parts. One is the isotropic component, which gives you a change in area, and the other one is an anisotropic component. So should I mean, is this clear, or I should explain it on the board. So I can show you with a simple example. The board is very nice. So think of a two-dimensional velocity gradient tensor. So I can decompose it Actually, I have another term in the decomposition which I have ignored, which was only these diagonal terms are 0 and the off-diagonal terms are the difference. For the moment, let's ignore that. Now think of a simple scenario where this xy or yx are I'm dropping off. So this first matrix, you see, this is the divergence of a velocity field, right? And this, any divergence, it gives you like a change in the area. Whereas this one, Vxx minus Vyy, you see this matrix, this is stressless. So this gives you any kind of anisotropic deformation. So if you have a deformation in the x direction, it will account for an equal deformation in the y direction. So any matrix, you can decompose it into an isotropic contribution and an anisotropic contribution, which is like the traceless symmetric part. So there is another contribution which for the moment I have ignored, and because the main deformation is happening along the x direction, we'll focus only on these components. Now let us look at the quantification. So as I said that the change in area is 0, and that one can see from this compression or expansion plot with time, which is roughly close to 0. And I will not go into these details. I will go to the main result. So this is the velocity gradient integrated over time. So what it should quantify? It should quantify how much the total blade has, length has changed. And we see, if you look at this number, it is roughly 0.2. So the total tissue has stretched by 20% of its original length. If I look at the average cell elongation, so remember the cell elongation was calculated locally for each cell, but now we are averaging over the whole tissue and calculating a single number with time. And if we look at it, we see that the average cell elongation finally at very large time, it is roughly 0.1, which means the cell at steady state has been extended by 10%. Now think of this. If you had a simple rubber band, which is made up of individual blocks, if you have some extension, you can expect that these individual blocks also has the same relative extension. But here it is not. We see a difference. Not only that, we see that the cell elongation with time, there was a non-monotonic behavior. It first overshoot and then coming back and going to a steady state or reaching some steady state value. So the question is, where is this difference coming from? No questions. So this difference comes from because this tissue, it is not a rubber band, it is not a passive material. There are activities which are happening at the level of individual cells, and that creates some more interesting things in this tissue, and this is topological changes or cellular rearrangement, and the idea is the following. So again, I try to give a geometric picture. So the idea is this whole network inside the cells, they can rearrange, they can exchange neighbor with each other. So you think of this. So you have two cells. If you now these dark blue cells, if you want to stretch them, these light blue cells will come close to each other and share a bond. Initially, these two cells were sharing a bond. Now, this will come and share a bond. And you can give rise to this kind of, because you have this whole active processes going inside each cell, there are stress build up within each cell, and the cell sometimes wants to release the stress by exchanging a neighbor. Okay, so there can be lot of these contributions. And you can also have this kind of contribution due to cell division and cell apoptosis. So what is the point then? The point is, if I want to write down a kinematic equation for tissue deformation, the rate of tissue deformation is contributed by the rate of cell shape change and these topological changes, which is purely due to rearrangements. So if it was, no rearrangement was there, it will be contributed by the change in the individual blocks. So the thing is, it is hidden here, but we actually have written down separately this contribution due to apoptosis and quantified from the experiment, but that change is not much as this whole developmental stage process. So when I show this, I have these results elsewhere. I can show you what was the contribution, but mainly here I will show purely the T1 transitions. But in principle, you can write down cell divisions, apoptosis, all of these, one after the other. So this is the, this one?
No, the shape where you define V tilde and Q tilde. Okay, maybe this one?
Yes.
So tissue shape changes V tilde and cell shape changes Q tilde.
Yes.
As in when you're making the triangles out of those polygons.
Yes. You
that-
are getting the full tensor,
All right.
and then you can break it up into this. So Q tilde is simply, I mean, if you are writing in tensorial notation, it is V tilde is basically so, in case this is what you are asking.
Maybe I'll ask later.
Okay. Okay, so now let me show you some examples. So again, let me reiterate. So the total deformation of this tissue will be a contribution because of how these individual cells, which it is made up of, changes on average, and how any rearrangement will contribute to this. So I think the following schematic will clear it. So always remember this equation and let us see. Suppose I don't have T1 transitions where they can rearrange, and my whole tissue elongates. So it elongates simply because of each individual cell elongating. What if these individual cells are not elongating, but the tissue is elongating? Can it be possible? Yes. And you see, I can rearrange the cells such a way that it extends in this region, but the cell's shape has not changed, but the overall tissue has become longer. Different.
Here we are already assuming that different cells have different shapes, right? Not like the.
No, this is just a schematic. All I'm saying is the cell shapes can change over time when subjected to stress. The boundary is also mechanically under tension. The boundary is not rigid. The boundary can also change. So I'm saying that this is one instance where the cells are not exchanging their neighbors, but overall, sorry, the cells are extending, there are no rearrangements, and that leads to a total change in this thing. On the other hand, I can have these cells, they are rearranging in such a way, this individual cell shape has not changed, but because of the rearrangement, because they are exchanging position, this has extended. I can also have a situation where the cell elongation is exactly cancelled by the deformation due to T1 transitions or topological changes, and that is this case, where the overall tissue has not changed. So you can have various such combinations. So the correct thing would be now to quantify each of these from the experiment and see what do we understand. So we have already quantified the V tilde and the Q tilde from the experiment. Now we can quantify this T tilde from the experiment. And how much time do I have left? 25 minutes, okay. So this is what each of this color corresponds to these different quantities. So the interesting quantity is the any kind of deformation or shear caused by these topological changes or rearrangements. So you see, initially there are two phases. So initially, the topological changes are negative. The shear caused by topological changes are negative, and at some point a transition happens where this topological changes becomes positive. Okay, so you can think of, if I think of this, so initially, because the way we have defined these tildes, all these tildes are in the positive x direction, a negative value will mean you have more elongation in the y direction. Okay.
I mean, here the transitions, I mean the T term, that's coming because of the shear and the cell arrangements, like we know from experiments, and it's just the sum, or it's individually quantified?
They are individually quantified.
Independently?
Independently,
okay.
Yeah. So I won't have time during the talk. Maybe after the talk I can show you how all these quantities are. The Q and the V is easy to see. The T is non-trivial.
Yeah,
I can show you
right.
later how they are quantified. Okay. So you can think that initially there are more T1 transitions in the y direction, and at some point it changes, and at the end there are more shear caused due to T1 transitions along the y direction. So here when we write T, it is the contribution to shear due to these topological changes. Okay, so because this is a kinematic equation, you are quantifying what deformation has happened, that you need to quantify. So let us see. So now we have this equation. Can we think of so we don't know what is the physics that is controlling tau tilde.
So I suppose you see these T1 transitions only in a growing tissue, right? In an adult tissue, when you stretch a tissue, the cells also get stretched accordingly, I suppose. You don't have cell rearrangements.
I think in principle, T1 transitions can also happen in like a tissue simply driven by thermal fluctuations. So it is like, if you can provide energy to these cells, which is sufficient to exchange a neighbor, squeeze through like a network of neighbors, that is essentially a T1 transition. So I think people have studied this kind of T1 transitions in like other networks also. Like in principle, for an equilibrium system also, just driven by thermal fluctuations, it can happen. But the contribution due to T1 transitions will be probably very less or negligible. But the whole idea is you have some network. Say think of like a pack of marbles or something. T1 transition is simply like one marble exchanging neighbor. So it is in some sense introducing some kind of fluidity. Because if you think how fluidity emerges in a material, it is because they are able to exchange neighbors.
Sure. So, but adult tissue wouldn't be so fluid, I suppose.
Probably
right?
not. I agree with you. So adult tissue is much more stiffer. So, but physically speaking, if you have, so for example, if you have cell division or cell death, that is also, cell death is something like a T2 transition. But yeah, I mean, if they're already like an elastic material, yes, but in principle, it can happen just driven by thermal fluctuations. That's what I'm saying. But I agree with you, mostly. Okay, so now we have this kinematic equation, but we also need to understand what gives rise to T. And here we have to introduce some physics because we don't know what exactly is, like we don't know in a, like the microscopic means of like what exactly gives rise to this T tilde. But what we can do is we can introduce something like a phenomenological equation, depending on what we think the factors on which T tilde can depend, and we can introduce some kind of a linear constitutive relation. And this is what we do here. So the first thing is we say that, okay, if we try to elongate, introduce cell elongation, if there are cell elongation happening, that can also change the T1 transitions. So it can depend on the T1 transition. And of course, to match the dimensions, because this is a rate, I have to put a tau here, which you can think like the time scale over which it's the relaxation time scale of the cell elongation. However, I can also have a contribution which is purely chemical in nature or active, meaning I know that the cell will have an actomyosin cortex, and if there is an isotropic distribution of proteins inside the cell, I can think of a scenario where this bond here contracts faster, and because of this contraction, these two cells comes close to each other. So this can also contribute to a T1 transition or the shear caused by T1 transition. So if this is the model, can we explain what we see experimentally? So let us see. So we have these two equations, but we have two equations and three unknowns. So what we'll do for the moment, we don't know how the flow field is generated so far. So we will take it as a fit parameter from the experiment and try to see if we can find out and if we can understand how the T1 transition behaves. So what we do, so this is our relationship. This is the T axis, this is the Q axis. We are plotting T versus Q. Now, what will be the behavior of T versus Q? It is linear, clearly, the way we have written the constitutive equation. So this will be something like this. If we plot the experimental data, it looks like this. So clearly something is wrong. However, if I now put some additional terms on the left-hand side, I see that I am able to match the profile very well. What I have done, we have introduced an additional time scale here, and this is a delay between T1 and Q. What it means is, basically I will explain in the next slide, that the moment you start changing the cell elongation, the T1 does not happen instantaneously. It happens with a delay. And that delay gives rise to this kind of a non-monotonic profile, and one can estimate those parameters from the experiment. And again, so what is the meaning of this delay? The meaning of this delay is the following. So you can think of this delay in terms of a response. Suppose you have the T1 transition depending on this Q. If you think that there is this function which introduces the delay between how the T1 responds to the cell elongation, then thinking of this equation, if this was instantaneous response, right? The T1 responds to the cell elongation instantaneously, then I should write this as a delta function, then I get back this original equation with which I started with. However, if I think that there is a delayed response, the simplest case is some kind of an exponential response, this exactly corresponds to the equation which we had written. So the bottom line is there is a delay between the T1 and the cell elongation, and now one can explain this a little better, which means that initially we have this negative T1 transitions, which means there are more elongation in the y direction, but because of the cell elongation which is increasing in the positive direction, at some point, because of the delay, the T1 responds to the cell elongation increase at a later time with a delay, and when it happens, it starts now increasing in the x direction. At the final time, what happens? The contribution to shear due to due to the cell elongation is exactly cancelled by the spontaneous T1 transitions which were happening in the y direction, which was clearly due to the active processes.
Is it within the error bars, or does that mean something? That I saw even in the previous one of the previous slides.
Yes, there is an oscillator.
No, in the beginning. So just from like if I look at the x-axis, exactly there, right there. If you look at the data, so before 20. So if you look at yes, exactly.
This oscillation.
Yes. Is it just error like within, because even that I saw even in the previous.
Yeah. No. It's the same plot. That's why you saw in the. No, but I don't know if it if one should if there is a significant oscillation at that time scale. But yeah, I don't know. At least the error bars are very small actually. So I have not plotted the error bars. It is there somewhere. But this there are error bars from 3 different experiments. So one can plot the error bar also, but I don't recall actually, to be honest. I think this is not, at least we didn't consider this oscillation. Okay, but overall if you look at the black curve, there is a damped oscillation and it is clear why it is there. Let me try to explain again. So now what is the effect of delay? So this is the actual curve, but if there was no delay, this is what it will look like. So you will completely miss this overshoot. Okay, and one can see now you will have discrepancies with the actual experiment here and here. And let us look at, so why we are getting this. So what has happened because of this delay, we have created a damped oscillator. So one can now write down, take these 2 equations, and you can see that the effective equation for Q is a second order damped oscillator. Okay. Now so far, yeah, so far we had been using this flow field from the experiment. If you don't put this flow field and just look for the purpose of like what these equations mean, this is what you will see. You will see several oscillations until it attenuates to a steady state. But when you introduce this flow field, it critically damps this to the steady state value. So what we have is like some kind of a feedback network between the T1 transitions and the cell elongation. So now, if you look at it, we have explained whatever we see in experiment and whatever we quantify, almost everything except for this last bit. We have not explained what is generating or giving rise to this flow field in the tissue which you saw right at the beginning. The cells were moving from the right to the left. And to do that, now again, so this is the difference. If you didn't have a flow field, your cell elongation with time will look like this. There will be a difference.
Somehow experimentally it's fine.
Can you? This is something intrinsic, which is probably some coupling with the individual cell polarities. So yeah, that will be an interesting thing to do. So what you are saying is, if one can identify the proteins which controls this delay between the, like in the response, and then tinker around with it and somehow regulate the delay, yeah. So I don't know. So okay. So let me come to the last bit. So what is giving rise to this flow field? And this again we have an hypothesis. The hypothesis is the following, related to what I earlier started with this actomyosin models, that I have these 2 pieces of tissue with a common boundary connected. If I think that this part of the tissue, which is the hinge, has more contractile stress, then this part, this can automatically generate a flow field. what in other words we are thinking of this. So I have these two parts of tissue, a very simplified rectangle model for this hinge and blade with a boundary. This part has more contractile stress, this part has less contractile stress, and this can generate a flow field.
So going back to the previous delay portion, so does the delay have to be implemented through the cytoskeletal network, or can it be like if let's say there is some force and then the force has to activate the resultant myosin proteins, if there is some feedback loop of that sort, will that give rise to a delay?
No, so we have not quite clearly identified the source of delay. What we have done from experiment is we have the data, we have quantified the shear caused by cell elongation, the shear due to topological rearrangements, and found that in between these two there is a delay. What is the actual biological control of this delay, that is still needs to be figured out. So, and that is definitely an interesting question, and that goes back to your question and also Sandeep's question.
So the cells are being marked by Catherine. That's the only output that you are
The
getting?
only output for us is we can resolve the mechanics of the individual cells. We can see how this individual cell shape is changing, how each cell is dying or contracting,
But
how they are exchanging.
at the same time, the cytoskeleton cannot be monitored, or has not been monitored in this set.
In this case, I don't know. Later, if there has been further studies.
I see.
So as I said, this was 2015.
A while back, yes.
So
Okay,
maybe there has been subsequent studies, but it will be a good idea to actually look at further studies. Now, this is of course a hypothesis that this part is contracting more than the other part. Oh, I am almost running out of time. And before I run out of time, I think my computer is going to run out of battery. So I didn't realize I came with full charge, but back? Oh, okay. Now, this is a hypothesis that you have more stress, contractile active stress here than here. So, that needs to be verified, and the way to verify is, first of all, we write down a constitutive relation, and again, this is a constitutive relation using like Onsager's reciprocity theorem. And so, the first part you already know. So, for any elastic material, one can write down this kind of a Hookean law, that the stress is proportional to the cell elongation with some coefficient. And the second term is very important, because this is saying that there is also, apart from the typical part which depends on the local elongation, there is also a contribution to stress because of activity, and this is due to actomyosin activity. And one needs to verify this from experiments, that it is indeed there. And how to verify that? And there are very nice experiments cell biologists do. So it is basically, you take the tissue, make a hole with a laser, and you see how this hole evolves. So basically, you calculate the overall deformation of this hole. It has two parts, the isotropic contribution, which is the change in area, and an anisotropic contribution. And the idea is, because creating the hole, you are creating flow fields, the stress is proportional to the flow field or the deformation, velocity deformation tensor. So you can plot the velocity deformation tensor. Let me show. So the experimentalists did this hole at various spots, and now plot, take all the data. Now this is our constitutive relation, which we have assumed that this is true. Because of the hole you created, it is proportional to the local velocity deformation or the recoil velocity. And if you plot this V tilde, which you can measure from the deformation of the holes, and the cell elongation, which we already know at the spots of the hole, we can plot this data, and we expect, because the sigma tilde is proportional to V tilde, it should follow the same linear relationship. Now, if there is an intercept, non-zero intercept, it means that zeta 1 is non-zero. There is an active stress within the tissue. So what one can do, one can compute the slope which will be related to the tissue elasticity. One can compute the intercept which will be related to the active stress. We cannot get the absolute numbers, but we can tell for sure that the ratio should be, whatever we get is correct. So now with this information, it seems that the model is correct. One last piece in the model, we are remaining with the boundary conditions. What are the boundary conditions? Again, we can do experiments. So it is now known that you have this wing and there is a outer extracellular matrix where there are polymer or filamentous networks which connects to the whole wing. So basically these acts like springs, and this tissue is under tension due to these spring connections. So how to verify that? So what one can do, so the springs, because they are not fluorescently labeled, they are in this dark region, you can make a laser cut there. And if you make a laser cut, for some reason this movie is not working, so I will play it. Oh, I cannot play. Okay, I will go to the next slide, maybe that will be. So if you make this cut here, you will see that this part of the wing, it shrinks faster because there are no boundary conditions. It's a free surface. It's a free, let's see. So if you make a cut here, so this is the wild type wing. Once you make the laser cut where you cut all the polymers which are connecting the wing, it shrinks further. If you make a cut here, it moves inside more. Now, the experimentalists, they also know the protein which actually regulates this polymerase network connecting to the wing, and it is called dumpy. So they can actually create a mutant of dumpy. And if you create a mutant of dumpy, you can see the difference. I will show you a movie. So this one is dumpy. So you can see there are no attachments, no springs attached, and it actually starts like moving very fast compared to this. So now with all this, we can put the boundary conditions also. We have large contractile active stress here compared to this, which we know from experiment. We put spring boundary conditions, we put spring boundary conditions. Now we have three different experiments, the wild type, the ablation where mechanically the springs were chopped off near here, the distal ablation where they are chopped, and the dumpy where there are no springs in the blade. We take all these different scenarios, we have 10 parameters but 15 independent curves. We calculate the force balance equation using all the set of equations we have. I will skip these equations, and the dotted lines are the fits to the experimental data, all fitted together, and you see that we kind of understand on average what leads to this kind of a deformation or like the shear due to these various quantities like T1 and cell elongation. I will quickly stop this part with one last movie, which is like, so our simplified model was this box, and which we put on top of this wing, and you can see how it looks like. Of course, we are not looking or trying to quantify the spatial pattern which comes out, because that requires more information from the experiments, which we don't have. We are looking at a very coarse-grained average cell elongation behavior, and it is able to explain that. But one can also look at the dumpy, which kind of gives you the same behavior. So, yeah, so what were the main observations? So first we saw that there is something called topological changes or rearrangements, which is a significant contributor to this overall deformation of this whole wing. And there seems to be a delay between the response of the T1 transitions to the cell deformation, and there are boundary conditions which actually are connected by like these polymers which controlled by this protein called dumpy. And this combined together, the active stresses and the boundary conditions can account for this overall tissue shape change. So How much? Do I have any time, or I should stop here? Okay, very quickly. So what I discussed mainly is a very old work where we were looking at the wing remodeling, and we had been recently looking at morphogenetic flows in the syncytial embryo in Drosophila. Now, I think there has been a I mean, Mithun has already introduced this formation of bicoid gradient during his lecture, right? So, and this is a very well studied morphogen gradient. So, in Drosophila, in the early syncytial embryo, so it is in the syncytial embryo, it is very peculiar, unlike many other organisms, because the cells are not completely formed. These are incomplete cells. They are not isolated from each other. They have furrows. There are partitions separating the nuclei, but the lower membrane is not yet formed. So there is a common cytoplasm. And this bicoid gradient, it is basically a very well-known, well-studied morphogen. It is deposited maternally at the anterior pole. This mRNA source is here for bicoid, and once you think that this protein is created here, it starts diffusing along this periphery. So inside, deep inside, it is like a dense network of yolk, so it cannot like move from here to here. So it diffuses along the along this periphery. And if one looks at the bicoid concentration and just projects this on the epi-axis, one gets this nice exponential gradient. And this is very important because probably Mithun has already discussed. So this provides a positional information to the target genes for the next developmental cycle, so namely for hunchback and Krupel. So they read this gradient length scale and express accordingly, and leads to differentiation of this whole embryo. Now the thing is, there has been several models. It has been a very controversial model because there has been several models proposed, and none of them actually is able to capture the whole, like all the features together, what is seen in bicoid gradient. The simplest one is this, and this is like time, you write the time evolution of this bicoid gradient, which has a source term at this anterior pole. There is a degradation term, and then there is a diffusion term. In this case, it is known there is no advection. Now, if you have such a term, it is clear that you have degradation, you have a source, and this competition between these two will give rise to a steady state profile, which is time independent. And the steady state of this is an exponential profile, where the length scale, the length of the gradient, is controlled entirely by the diffusion coefficient and this degradation rate. Now the interesting thing is, it has been observed that this length scale actually scales with the embryo length. So in some of the embryos which are bigger, this length scale is also bigger. But if you go by this model, there is no way to control, like scale this length scale. So this is really like an interesting question, that this model is not sufficient. And what we started looking at is the aspect that although we introduce a simplified model of diffusion, these individual regions around the nuclei, this is not like a free space. There are lot of network, there is the presence of the cytoarchitecture. There are these microtubule networks which becomes different during interphase or metaphase, where the spindles flip. And in this case, because of this kind of crowding and this kind of a heterogeneity, this heterogeneous environment can become important, and then there can be nuclear shuttling and all. So I will not have time, I will skip this. So we did a recent study to look at a membrane bound protein, how it diffuses in presence, which can also bind to the membranes. But the main idea is, let me skip quickly, that cytoarchitecture plays a crucial role in shaping up the steady state gradient. And so one should consider heterogeneous diffusion, like kind of fast slow diffusion, and also nuclear shuttling. And the question is, if you combine this, can it explain this scaling with the embryo size? So this is one problem which we have already published, but the recent problem which we had been studying is another fascinating problem, where you see there are these dark spots which are the yolk particles, and you are looking at the syncytial embryo, and as time progresses, these dark spots are segregated actively from the more periphery towards the like towards the inside, near the bulk. And this segregation is an active segregation, namely this microtubule network assists this. So we are trying to develop, use a combination of data analysis and hydrodynamic model to understand, like, if we can explain this kind of a segregation. So I will skip all these details. So let me acknowledge the people who were involved. And I didn't really talk about the second work, but yeah, so I think I should stop here. Thanks for your attention.
Okay, there were quite a few questions during the talk, but if there are one or two, immediate questions, we can take them. Okay, if not, we can take it during coffee. I think Amitabh will be happy to answer any questions you have during coffee. So let's thank Amitabh once more, and coffee is right outside.